Assalamualaikum warahmatullahi wabarakatuh. Today, we will present an article titled Inhibitory Effects of Lactobacilli of Goat's Milk Origin Against Growth and Biofilm Formation by Pathogens, an in vitro study. Significant discoveries of this article are Pseudomonas aeruginosa and Staphylococcus aureus are bacteria that form biofilm and were responsible for posing significant challenge to the food industries, causing the contamination and equipment damage. Probiotic lactic acid bacteria (LAB) found in the dairy products like goat milk have offered a natural alternative for inhibiting the biofilm formation and controlling pathogen in the food industry. This study aimed to evaluate the antimicrobial and antibiofilm potentials of goat milk lactobacilli against the Pseudomonas aeruginosa and Staphylococcus aureus. The hypothesis suggested that lactic acid bacteria (LAB), lactobacillus species from the goat's milk, played a role in manipulating the level of antimicrobial activity and biofilm growth of two pathogen strains, Pseudomonas aeruginosa and Staphylococcus aureus. The objectives are, first, to compare the antimicrobial activity of the CFS extracted from the each lactobacillus strains against the Pseudomonas aeruginosa and Staphylococcus pathogen. Second, to investigate the inhibitory effects of the CFS and also HCFS of the lactobacillus strains isolated from the goat's milk on the biofilm formations of the Pseudomonas aeruginosa and Staphylococcus aureus. In this case study, among the method used is, first, for the antimicrobial activity, it will be using the agar diffusion assay, method on the cell-free supernatant extract from the LAB strain. This can help to produce Pseudomonas aeruginosa and Staphylococcus aureus inhibition zone, where provide information of the varying degrees of activity. Second, in vitro method using a microtiter plate will be used in order to investigate the inhibitory effect of the CFS lactobacillus species, and then this technique is used as the stepping stone to calculate the biofilm biomass by measuring the intensity of the crystal violet using the UV spectrophotometer. Biofilm detection uses the crystal violet because it is low cost, simple technique, and also high reproducibility. And third, light microscopy used to show the clear difference in biofilm structure between the treated CFS and also the untreated CFS controls. Next, I will pass to the Azhani for the details on the result and also discussion.
Thank you, Izzah. Moving on to the results and discussion. Several LAB strains have been shown to possess antimicrobial activity towards P. aeruginosa and S. aureus, with the highest inhibitions of growth activity are demonstrated by the LAB strain GM12, 13, 15, 17, and 18 with 20 to 24 mm of zone of inhibition. The effects of LAB strain GM12, 13, 15, 17 as well as N and HCFS has been shown to significantly inhibit biofilm formations of the two tested species. It was suggested that the bioactive peptide and bacteriocin potentially inhibit the formations of biofilm by these pathogens, and the inhibitions found to be time-dependent and varied within the strain. Additionally, reductions with biofilm suggest that CFS interfere with the initial stage of attachments and promote detachments at later stages. Next, microscopic images illustrated that the inhibitions of biofilm formations by lactobacillus CFS by the treatment with GM12 resulted in scatter cell growth due to the decrease of microcolony formations. The results suggested that the inhibitions of biofilm was primarily caused by the LAB strain. However, there may be an identified antimicrobial compound that might inhibit the biofilm instead. Some LAB strains may also show higher inhibitory activity under different conditions. I pass the presentation to Husna.
Thank you, Azhani. We can conclude from this article that lactobacillus strains isolated from goat's milk samples has exhibit significant antibacterial effects and biofilm inhibitory activity against Pseudomonas aeruginosa and Staphylococcus aureus, suggesting their potential use as biocontrol agents combating bacterial pathogens. I agree with the conclusions drawn by the authors based on the presented data and valuable insights in discussion regarding the probiotic potential of these lactobacillus strains. The authors did consider supporting evidence from previous studies on the effectiveness of antibacterial properties in CFS lactic acid bacteria against Pseudomonas aeruginosa and Staphylococcus aureus. To strengthen their own findings, the authors cited additional external sources confirming this antibacterial activity and its cause against the same two pathogens, and even adding a third pathogen, Bacillus subtilis. The first limitation is authors use references that are more than 5 years old. Although research may be outdated, use less reliable methods and not reflect the current state of knowledge. Secondly, the study scope seems limited to two specific pathogens only, Pseudomonas aeruginosa and Staphylococcus aureus. Thirdly, the research was conducted in vitro, which may not fully represent the complex interactions that occur in vivo. That's all from us. Thank you so much for listening.